Why is DNA a double helix? Chapter 1. The necessity of the twist. Ask a child to draw life's code, and you'll get the same answer you'd get from a Nobel laureate's doodles on a dinner napkin. A ladder, tipped on its side, spiraling up and up. The double helix has become an icon so familiar it risks seeming inevitable, like the roundness of planets or the blueness of a clear sky. But nothing in chemistry is inevitable. Shapes are bargains struck between atoms and water, between charge and geometry, between the mathematics of probability and the stubbornness of matter. So, if we're to answer the question honestly, why DNA is a double helix, we have to follow those bargains from their first handshakes to their surprising consequences. Begin not with DNA, but with water. Biology is an art that happens in a solvent, and water is not a passive stagehand. Its molecules are tiny magnets, with rules about who they'll touch and how they'll spin around each other. They favor partners who present charges and polar edges. They shun large, flat, oily faces. Down at the scale of a nucleotide, the building block of DNA, every decision about shape is made in conversation with those preferences. A nucleotide has 3 parts: a sugar, deoxyribose, a phosphate group, and a nitrogenous base. The phosphate is an emphatic entity, bearing a negative charge that will, in an aqueous environment, pull a countercloud of positive ions around its sodium, potassium, magnesium waiting like peacekeepers between repelling neighbors. The base is a flat aromatic ring or two. Adenine and guanine are purines, 2 rings. Cytosine and thymine are pyrimidines, 1-ring sheets of electrons that detest the pushy attention of water. The sugar is the bridge, a 5-carbon ring with a personality of its own, chiral and constrained, holding the phosphate and base at particular angles. Already you can sense tensions. A hydrophobic base wanting to be hidden from water, a hydrophilic, negatively charged phosphate wanting to remain exposed so the solvent can stabilize it. A sugar with a fixed handedness and a particular set of links to offer. Put 2 such nucleotides next to each other, and they will respond to water's demands. Tuck the oily bases into the interior, leave the charged phosphates in contact with the solvent, and let the sugars stitch the arrangement together. The easiest way to accommodate both needs at once is to build a ladder where the rungs are the bases and the rails are chains of sugars and phosphates. But ladders don't thrive in water. They twist. Why the twist? Because twisting trades one kind of energetic cost for a more favorable set of gains. If you kept the ladder flat and straight, vast surfaces of the bases would remain exposed to water, and the negative charges along the rails would line up to glare at one another, each phosphate repelling the next. Rotate the rungs just a little and the bases stack like coins, reducing their area of contact with water and allowing their electron clouds to sit over one another in a stabilizing dance known as pi stacking. Meanwhile, the rails can step aside from perfect alignment, reducing direct charge-charge repulsion. The twist is an optimization. It increases favorable base stacking, decreases unfavorable phosphate repulsion, and satisfies the geometric constraints imposed by the sugar. The helix is chemistry's compromise that became life's triumph. Pull the camera in closer. Within this twist, each rung is not just any pair of bases, it is a complementary pair. Adenine prefers thymine, guanine prefers cytosine. These preferences are not arbitrary romances, but the bookkeeping of hydrogen bonds and shape complementarity. Hydrogen bonds are modest attractions, hydrogen sharing between donor and acceptor atoms, strong enough to confer specificity, weak enough to let the strands come apart when work must be done. A and T make 2 such bonds. G and C make 3. But the idea that hydrogen bonds alone dictate pairing would be a half-truth. The other half is shape. The AT pair and the GC pair are nearly the same width. This uniformity is crucial. It keeps the rails, the sugar-phosphate backbones, at a consistent distance, preserving the helix's symmetry and enabling the elegant zipper logic by which information is copied. Those hydrogen bonds do more than hold the rungs together. They dictate the grooves, the major and minor grooves that run like highways along the outside of the helix. Because the base pairs are not centered perfectly between the rails and the glycosidic bonds, the bonds from each base to its sugar arrive at a slight angle. The helix presents 2 kinds of furrows. The major groove is wide, with base identities readable from the outside. The minor groove is narrower, with subtler cues. Proteins evolved to recognize patterns of hydrogen bond donors and acceptors exposed in these grooves, skating along the DNA and stopping only where the pattern matches their mission. Without the twist, the grooves would be different or absent, and the language of recognition would be harder to write. Now glance again at the sugar. It is deoxyribose, not ribose, and it's a specific kind, deoxyribose, a chiral molecule that cannot be superimposed on its mirror image. This handedness imprints itself on the polymer, link many sugars and phosphates together, and the geometry of each bond, the preferred angles between atoms, nudges the growing chain toward a right-handed spiral known as B-form DNA under typical cellular conditions. The sugar pucker, the particular way the sugar ring adopts a non-flat conformation, and the orientation of the glycosidic bond constrain how each base can stack on the next. Given the choices offered by terrestrial chemistry, the path of least resistance is a right-handed double helix. In drier conditions or when the water activity changes, the helix can adopt the A form, still right-handed, tighter and more compact. The form RNA prefers. Under certain sequences and high salt conditions, a left-handed form, zDNA, appears, a reminder that even icons have exceptions when the environment tweaks the energetic balance. Electrostatics adds another layer to the story. The backbone's negative charges would repel one another enough to blow the structure apart were it not for counterions and proteins like histones. In eukaryotes, DNA wraps around histone octamers, forming nucleosomes. The double helix has a pitch and diameter that allow precisely 1.7 turns to be arranged around a histone core, with about 147 base pairs making a neat spool. This is not an accident. The mechanics of bending a double helix, its persistence length, a measure of stiffness, and the periodic exposure of the minor groove line up to make such wrapping energetically plausible. A flat ladder would buckle and kink. A twisted uniform cylinder bends predictably. The helix is not only a stable storage shape, but an engineer's dream. It manages in the same motif to be stiff enough to maintain integrity over short spans and flexible enough to compact into meters of chromosomal architecture. Consider now information and error. Life is brittle without faithful replication, and yet life is impossible without change. The double helix solves both needs by copying complementarily. Each strand carries enough information to reconstruct its partner. Heat the system or invite an enzyme called helicase, and the hydrogen bonds unzip. DNA polymerases go to work, reading exposed bases and writing new ones in complement. Complementary base pairing is not just a trick for stability, it's the reason copying can be as simple as make the opposite. The twist assists here, too. The geometry of the base pairs in the helix places the 3-hydroxyl group of the sugar where new nucleotides are added at consistent intervals and orientations. Polymerases evolved to fit the helical duplex like a hand fits a familiar tool, their active sites keyed to the B-form's parameters. An entirely different geometry would have demanded an entirely different suite of enzymes. Perhaps life could have found another path. But the double helix made one path exceptionally smooth. The helix also offers error signals. If a wrong base slips in, the local geometry is subtly wrong. The pair is too wide or too narrow. The hydrogen bonding pattern is off. The stacking is disrupted. Polymerases feel the misfit. Proofreading domains back up and replace the errant nucleotide. Mismatch repair proteins patrol after replication, feeling for bumps in the otherwise uniform road. The same shape that enables recognition by transcription factors also allows recognition of mistakes. Structure begets function, and uniformity begets a way to detect deviations from uniformity. Now, imagine alternative worlds. Could life have used a triple helix? Chemistry answers, in principle. Yes, there are triplexes known as hdNA in certain sequences, and DNA can form G quadruplexes in guanine-rich regions, and I motifs in cytosine-rich stretches under acidic conditions. But those are local, special case structures with roles in regulation and genome architecture. They are not global solutions to storage and replication. Triplexes would compress sequence possibilities, complicate base recognition, and burden replication machinery. A single-stranded genome, many viruses manage it, but at the cost of stability and error rates ill-suited to large, long-lived organisms. A helix of more than 2 strands is too rigid and complex. Fewer strands are too floppy and error-prone. 2 turns out to be a sweet spot for stability, specificity, and economy. A Goldilocks choice reached not by deliberation, but by the quiet arithmetic of energy minimization and evolutionary success. History, too, gives us a clue. Before DNA claimed the throne, RNA likely ran a smaller kingdom, the hypothesized RNA world, in which carriers of information also acted as enzymes. RNA almost always adopts an A-form helix in duplex regions, a consequence of its 2-hydroxyl group and the way ribose puckers. That helix is thicker and shallower, the major groove deep and narrow, the minor groove wide and shallow, a geometry that makes certain kinds of recognition challenging. DNA, by losing the 2-hydroxyl, hence deoxy, gains chemical stability, fewer accidental hydrolyses, and a B-form helix whose major groove is open enough to read without unzipping. In a world growing more complex, DNA's double helix proved a better long-term library, while RNA pivoted to roles as courier and catalyst. The high school diagram of the central dogma, DNA-RNA protein, hides a structural truth. Each arrow implies a shape that allows the handoff. There is also the matter of chirality writ large. All terrestrial DNA uses D sugars and L amino acids in proteins. This homochirality lets polymers pack and interact consistently. A right-handed B-form helix presents a predictable face to proteins built of L amino acids. The 2 chiral worlds evolve together like glove and hand. Would life elsewhere choose the opposite? It might. The mathematics would be the same with signs flipped. The important thing is not which hand the helix takes, but that it takes one hand and keeps it a continuity that preserves compatibility. The double helix is also a mechanical solution to the problem of long threads in small rooms. Twisting introduces topological properties linking number, twist, writhe that can be managed rather than merely suffered. Topoisomerases cut and rejoin strands to relieve supercoiling, resolving knots and tangles that would paralyze replication and transcription. The helix gives biology a dial to turn, over-twist to compact, untwist to open. Chromatin architecture, from loops to TADs, topologically associating domains, from enhancer-promoter kisses to the choreography of whole chromosomes during mitosis, builds upon this manageability. A rope that can be twisted and untwisted can also be stored and deployed. A flat ribbon resists tidy policies. All of this waters tastes, base stacking, hydrogen bonds, sugar geometry, electrostatics, groove readability, homochirality. Topology answers the why, with the patience of many small reasons adding up. But perhaps the most profound reason is this: the double helix makes life legible to itself. A genome is not only a hoard of symbols, it is a surface covered in signs. Proteins read promoters because the major groove displays a code in the language of chemistry. Donors and acceptors, methyl groups and polar edges in patterns specific to each base pair. Methylation, a tiny chemical tag placed by enzymes, slightly changes that surface, tweaking who can read and who cannot. Damage produces other marks, oxidized bases, ultraviolet dimers that call for repair. Epigenetics, regulation, stress responses, all begin as edits to a surface already rich with meaning because it is twisted in just this way. One can imagine, in a thought experiment, a straight ladder where base identity is hidden from the outside. All recognition would require unzipping. Every conversation would begin with a shout. The double helix is quieter. It allows whispers through the grooves, quick touches and no-go decisions, a cell's equivalent of glances across a room. The helix is not only an energy minimum, it is a communication protocol. And still, the helix would be a mere curiosity if it were not also copyable. Complementarity ensures duplication. Stacking and grooves ensure endurance and legibility. Chirality and uniformity ensure machinability. It is a vast collaborative design convened by physics and perfected by evolution. Even the exception Z-DNA zigzags in high salt stretches, G quadruplexes crowning telomeres, serve rather than undermine the central motif, adding local variation where regulation demands nuance. If you feel that the answer is both satisfying and incomplete, you're hearing the right echo. Chemistry supplies the constraints. Evolution supplies the selection. DNA is a double helix because, among the structures consistent with those constraints, it was one that could be built by small steps from simple parts, one that could be recognized and copied by molecules flexible enough to evolve, one that could be folded into cells and bodies and species, then written into the long story of Earth. It is shaped the way rivers shape valleys, not because some general decreed a path, but because drops of water, faced with gravity and stone, found the route that could carry them all the way to the sea. There is also the grace note of beauty, the sense that when we pull the strands apart and read their sequence and then close them again, we are handling not just a molecule, but a history. The helix insulates and exposes, hides and reveals. You can tug it open to copy and then let it reseal seamlessly. You can wind it around proteins and still leave points of access. You can mark it without damaging it and erase the mark when the message is no longer needed. Form and function have struck their bargain so cleanly that, once you understand it, it is hard to imagine any other outcome. Yet this inevitability is backward-looking. The past necessities always look faded. So why is DNA a double helix? Because water preferred it, electrons favored it, sugar angles required it, ions permitted it, and evolution exploited it. Because 2 is the number that lets a code be both stable and copyable, readable and repairable. Because the helix's twist makes it compact and cooperative, a thread that can be knotted and unnoted without breaking. Because molecules that could have been shy and chaotic found, in the twist, a way to be both guarded and gregarious. We might end, for now, with the image of a cell dividing. Chromosomes line up and are tugged toward opposite poles. The double helix in each splits its 2 strands, and polymerases race along, writing complements, converting possibility into near certainty. The twist, there is not decoration. It is the unwritten rule that allows the whole scene to proceed. The question, why, turns out to be answered not by one principle but by a chorus singing in tune: stability, specificity, machinability, readability, topology, evolvability. You hear the harmony most clearly when you see the helix not as a symbol but as a solution. The spiral staircase is simply the most graceful way, under the known laws of chemistry, to climb from atoms to meaning. Chapter 2. What the spiral makes possible. There is a moment every experimentalist secretly hopes for. You draw a line through scattered data points, and suddenly the line tells a story larger than the scatter. The double helix is that kind of line. We can list the chemical reasons for the twist water's preferences, base stacking, electrostatics, and feel satisfied. But the shape also organizes consequences, as if the geometry itself wrote a to-do list for life. This chapter is that list. What the spiral makes possible, how cells exploit it, and why the same logic underlies copying, reading, repairing, packing, and evolving. Begin with a practical number. About 10. 5 base pairs per turn in B-form DNA. A rise of roughly 3.4 angstroms per base pair. A diameter near 20 angstroms. These aren't trivia. They're the dimensions that proteins have evolved hands to fit. Polymerases, helicases, transcription factors, restriction enzymes, nucleosomes, each is a tool shaped by those measurements, the way a craftsman's chisel is shaped by the hardness of wood. If the helix were tighter or looser, the tool chest would be different. But given this pitch and width, life built a workforce that can work quickly and with astonishing fidelity. Consider Chargaff's rules: adenine equals thymine, cytosine equals guanine, an empirical whisper that preceded the double helix. The moment Watson and Crick placed base pairs inside the rails and saw those ratios fall out as necessity, chemistry, and information married. The AT and GC pairs are not just equal on average, they are almost equal in width, so the rails stay equidistant. That uniformity is what makes the helix look like a well-machined part. If A occasionally paired with G and T with C, the staircase would wobble, and proteins tuned to the expected geometry would stumble. Specificity and uniformity reinforce one another. The right pairs both fit best and keep the whole structure evenly spaced, and that evenness is readable. Readable, this is crucial. stand on the outside of the helix where proteins stand. The major groove exposes a chemical barcode for each base pair. Patterns of hydrogen bond donors and acceptors, methyl groups and polar atoms laid out like flags. The code for AT differs from TA, GC differs from CG. In the minor groove, the patterns are less distinct. This asymmetry invites a division of labor. Proteins that must recognize specific sequences without prying the helix open. The transcription factors that decide which genes should speak. The restriction enzymes that target precise motifs read the major groove. Others, concerned with local shape more than precise letters, probe the minor groove and the way it narrows or widens with sequence. DNA is not just a string of symbols. It is a surface embossed with meaning, and the twist is what arranges the embossment into two walkable lanes. Now, imagine a polymerase in motion, copying a strand. The enzyme clamps around the duplex like a protective hand, feeds the template through a channel, and checks each incoming nucleotide against the current base. Geometry is the judge. Correct pairs snap into a near Watson-Crick shape that positions the 3-hydroxyl to attack the incoming phosphate chemistry made precise by alignment. Wrong pairs misalign, slowing the reaction enough to invite proofreading. Many DNA polymerases have an exonuclease domain that backs up and excises mistakes. After replication, mismatch repair teams patrol the fresh roadbed, feeling for bulges and dents that betray mispairs. The double helix makes all this possible because it is consistent enough that deviations stand out. In a world of varied geometry, wrong would be harder to define. In a world of single strands, error would hide more easily because local shape shifts constantly with solvent and temperature. The same uniformity aids chemical triage. Different kinds of damage advertise themselves in different ways. Ultraviolet light can force adjacent thymines to dimerize, kinking the helix. Base oxidation introduces chemical features that repair enzymes sniff out. Alkylation decorates bases with groups that don't belong. Double-strand breaks present a catastrophic discontinuity. Nucleotide excision repair recognizes distortions in the helix's shape and cuts out a patch. Base excision repair pinches out individual damaged bases. Non-homologous end-joining welds breaks with haste. Homologous recombination borrows information from an intact copy to repair with care. All of these are shaped around the expectation that a stretch of undamaged B-form DNA is a smooth, predictable cylinder with regular grooves. Repair is the art of noticing the irregular, and regularity is what makes irregularity shout. The helix also simplifies decision-making for proteins that must bind and unbind rapidly. Suppose a transcription factor needs to locate a 6-base motif among millions. It cannot afford to sample every site exhaustively. Many DNA-binding proteins use a combination of 3-dimensional diffusion and 1-dimensional sliding along the helix, intermittently hopping off and reattaching, spiraling along the grooves while maintaining a loose contact. The groove geometry provides a track. Electrostatics helps too. The negative backbone invites positively charged patches on proteins, creating long-range attractions that guide the first encounter and permit rapid scanning without getting stuck. The helix's topology, its continuous, regular spiral, makes this kind of facilitated search possible at biologically useful speeds. Topology adds more than a track. It adds levers. A double helix has a linking number how many times the two strands wrap around each other. Because the strands are closed or effectively anchored during many processes, changes in twist often convert to writhe, coiling the helix upon itself. Cells exploit this. Bacteria maintain DNA in a state of negative supercoiling, which makes it easier to open local regions for replication or transcription. DNA gyrase, a topoisomerase, introduces negative supercoils. Other topoisomerases relax or redistribute them. In eukaryotes, nucleosome wrapping and higher-order chromatin architecture store and release superhelical tension during transcription, helping polymerases plow forward like engines moving through a landscape of hills and valleys, the terrain shaped by twist and writhe. The point is not merely that DNA can be twisted, it's that the double-stranded topology turns twist into a handle life can grab. Packaging the genome at human scales turns handles into machines. Picture the classical hierarchy: helix, nucleosome, chromatin fibers, loops, domains, chromosomes, not as a neat stack of boxes, but as a dynamic scaffold that breathes. Each nucleosome is an octamer of histones, 2 each of H2A, H2B, H3, H4, with DNA wrapped 1.7 turns around it. The path is not arbitrary. It places minor grooves in contact with histone surfaces at periodic intervals, reflecting the helical repeat. Sequences with T-rich motifs, every to 10 base pairs, bend more readily to wrap. Linker histone H1 binds the entry, exit DNA, adjusting the angle and aiding compaction. Remodelers slide or evict nucleosomes to make room for polymerases. Writers and erasers add or remove chemical tags on histone tails, acetylation, methylation, phosphorylation, tuning how tightly DNA associates. All of these interactions depend on DNA presenting the right thickness, stiffness, and groove positions, the givens of the double helix. Stiffness matters numerically. The persistence length of bDNA is about 50 nanometers, roughly 150 base pairs. Below that scale, the helix behaves like a relatively rigid rod. Above it, it bends and coils. This balance allows the fiber to be both structured and flexible. Were DNA much floppier, local regulatory geometry would be too noisy. Were it much stiffer, packing would be too costly. The double helix, with its base stacking and phosphate backbone, sets this stiffness at a sweet spot for life-sized as life is. Even the exceptions decorate rather than erase the rule. G-rich sequences can form G-quadruplexes, stacks of guanine quartets stabilized by monovalent cations, particularly near telomeres or promoters. C-rich regions can form I-motifs under acidic conditions. RNA, DNA, hybrids, R-loops, naturally form during transcription, with the RNA adopting A-form geometry and the displaced DNA single strand left vulnerable or ready to signal. zDNA, left-handed and zigzagging, appears under torsional stress or in special sequences. Each of these structures acts like a punctuation mark in the sentence of chromatin, but the sentence is still written in B-form words. A library cannot be useful if it cannot be opened and read, and reading requires localized unzipping. The helix excels at controlled opening because hydrogen bonds between base pairs are strong enough to secure, weak enough to yield. GC-rich regions resist more. AT-rich regions melt more easily. So, promoters and origins of replication often bias for AT where opening is wanted. Helicases pry strands apart, ATP-powered wedges that exploit the directional asymmetry of the backbone 5'3" and 3/5", and the repeat of the helix. Single-stranded binding proteins coat the separated strands, preventing hairpins. Primuses lay down RNA primers, polymerases extend, sliding clamps keep them close, ligases seal nicks. The dance is choreographed around a geometry that is regular, directional, and separable. Double-stranded but unzip-ready is not a trivial property. It is the reason copying and transcription can be both fast and accurate. Accuracy is not perfection. Tautomers, the rare, transient rearrangements of base protons, can trick polymerases. Chemical insults slip past repair. Breaks occur in replication forks and under stress. Here again, the helix's uniformity saves the day after it has created the problem. Mismatch repair detects and fixes mispaired bases by reading shape. Homologous recombination uses the fact that two helices with the same sequence can align and exchange strands, a feat of complementarity that would be far more difficult if sequence identity didn't translate into a predictable shape. When chromosomes pair in meiosis, a similar logic lets homologs find each other among crowded nuclear space. They don't just match by content, they match by how content shapes the helix. The double helix also enables a quiet third axis of writing, epigenetics. DNA methyltransferases add methyl groups to cytosine, often at CpG dinucleotides, modest decorations that do not change the base pairing code but alter the major grooves pattern, silencing or promoting binding by specific proteins. Hydroxymethylation and further oxidation steps add nuance. Binding proteins with methyl reading domains respond accordingly. Histone modifications did not have to interface cleanly with DNA shape, but they do. The helix's grooves admit histone tails and readers at the angles needed. Epigenetic marks would be far less useful if visibility from the outside were poor. The fact that the helix advertises base identity means that small chemical edits change the advertisement in ways readers can detect without ripping the page. Tell part of the story through history, because history reveals design constraints through human struggle. Rosalind Franklin's X-ray diffraction images of DNA fibers, Photo 51, most famously showed a cross-pattern diagnostic of a helix, layer lines that marked the 3-4 angstrom repeat, a strong meridional reflection announcing the 10-ish base pairs per turn. Those images were not just pretty. They quantified the structure in a way that left few plausible alternatives. Maurice Wilkins, Raymond Gosling, and colleagues contributed, as did the chemical intuitions of Linus Pauling and the base pairing insight sparked by model building. Chargaff supplied the base ratios. Jerry Donahue reminded colleagues of the correct keto forms of the bases. Oswald Avery had shown DNA carried heritable information years before. When the model clicked, it clicked because the geometry answered all the prior facts at once. The why was baked in. A double helix satisfied physico-chemical demands and encoded complementarity required by information. Let's walk outward from the molecule to the cell cycle. Before a cell divides, it must replicate its DNA. Multiple origins fire in eukaryotic chromosomes. Forks proceed bidirectionally. Replication timing coordinates with chromatin state. Topoisomerases prevent torsional stress from stalling forks. Cohesins hold sister chromatids together. Checkpoints pause the cycle when damage is detected. The elegance of the scheme depends on the double helix being a discrete object with defined ends and a predictable behavior under force. During mitosis, condensins compact chromosomes. Microtubules tug on kinetochores. Segregation splits the copied helices into daughter cells. Every step assumes that DNA is sufficiently robust to be pulled, wound, and unwound without losing registration, that a duplex remains a duplex under load, that unzipped regions can rezip without misaligning. A different geometry would make these feats harder, or would require a different set of proteins and checkpoints evolved from scratch. Zoom into bacteria for contrast. No nucleus, no histones in the canonical eukaryotic sense, though histone-like proteins exist, but the same logics apply. Negative supercoiling stored in the chromosome aids opening. Nucleoid-associated proteins shape domains. Replication proceeds from a single origin. Transcription and translation can be coupled. CRISPR, the adaptive immune system co-opted for genome editing, uses guide RNA to form an RNA-DNA hybrid at target sequences and the enzyme's hands to interrogate the duplex. Recognition relies on local unwinding around a short motif, PAM, that signals "try here." Again, the helix's rules, how easily it opens, how it presents information externally, set the energy barriers for recognition and cleavage. Engineering with CRISPR works because the natural system reads a structure we now understand well enough to direct. Synthetic biology tests the boundaries of why. Researchers have added extra letters to the genetic alphabet, unnatural base pairs, showing that, within limits, the double helix tolerates new shapes so long as stacking and pairing remain compatible enough not to break the helical geometry. Others have built Hachimoji systems with 8 building blocks in vitro. These are proofs of concept, not rivals to life's DNA, but they tell us something. The double helix is a generous host. The precise chemical identities of the bases can vary if the bigger picture constraints stacking energy, uniform width, complementarity that preserves geometry are met. That generosity hints at the deep reason again. The helix is a solution space, not a single point. Life happened to settle on A, T, G, C, but it chose so that the helix's demands were satisfied. Change the parts carefully, and the shape still works. Packaging at the genome scale is an architecture problem with helix as its brick. Loop extrusion by SMC complexes, cohesin, condensin, forms chromatin loops and topologically associating domains, TADs. Enhancers communicate with promoters over distances by looping. Insulators block crosstalk. The energies involved are measured in units of thermal fluctuations, which means small differences in bending and twisting rigidity set by sequence and helical shape matter. AT-rich sequences bend differently than GC-rich. Phased motifs can pre-curve DNA to favor or disfavor nucleosome occupancy. This is indirect readout. Proteins sense sequence by sensing how sequence shapes the helix's mechanics. Again, shape transduces letters into forces, and the double helix is the transducer. The helix even regulates time. Telomeres, the protective ends of linear chromosomes, use repeats that fold into structures, including G quadruplexes and shelterine complexes that manage when ends are accessible to telomerase or repair factors. If ends were chemically indistinct from breaks, repair would mistake them and fuse chromosomes. If they were too alien, normal replication would fail. The double helix allows ends that are the same kind of material as the rest, but dressed differently enough by sequence and bound proteins to signal "do not join." The same geometry underlies recombination in meiosis, where homologous chromosomes exchange segments. The pairing requires alignment of helices at many points. Shape matching in 3D makes a search in 1D sequence tractable in the crowded 3D of the nucleus. Think about energy bookkeeping. Stacking interactions between bases are the primary stabilizing contribution to the duplex, with hydrogen bonds providing specificity and supplemental stabilization. That priority order explains why mismatches, even when hydrogen bonds can form in rare tautomeric states, often destabilize strongly. Stacking is disrupted. It also explains why intercalating agents, flat molecules that slide between base pairs, can cause frame shifts or inhibit replication. They bribe the stacking system with counterfeit coins, widening the helix and confusing polymerases. The helix is stable because coins stack neatly. Anything that perturbs the pile has outsized effects. Chirality we met in Chapter 1. Here we see its consequences in proteins that follow the helix's paths. Many DNA-binding helices in proteins, alpha helices, insert into the major groove with a geometry that complements right-handed bDNA. That is not a coincidence. With L-amino acids forming proteins, alpha helices wind right-handed. The major groove's dimensions suit these helices, making side chain presentation optimal for reading base patterns. The glove fits the hand because both evolved together. Swap chiralities, and the complementarity would persist with mirrored tools. This symmetry in logic, not in matter, reassures us that the Y is not earth parochial. It is physical. What about the claim that life could have chosen a different path entirely, no helix at all? In principle, other polymers could store information. Peptidic nucleic acids, PNA, threose nucleic acids, TNA, glycol nucleic acids, GNA. Many of these form duplexes with DNA and RNA, but notable is how often helices reappear. Stack flat units, place repeat charges on a backbone, and ask water to be the solvent. The minimum energy solution looks like some kind of helix because twisting reduces exposed hydrophobic area and distributes charge. It is not that a helix is inevitable in all conceivable chemistries. It's that, in the chemistry we inhabit, aromatic bases in water, charged backbones in a salty world, a helix is a very good answer. Double strands then buy the miracles of complementarity and easy copying. The path of least free energy is also the path of greatest utility. Utility scales from molecules to meaning. The central dogma, DNA to RNA to protein, rides on reversible and irreversible steps. DNA's double helix is relatively inert, protected. RNA's A-form helix is more reactive, able to fold into catalysts and adapters. Proteins are the most varied in shape and chemistry. That gradient of stability and reactivity isn't ordained by philosophy. It is written in sugar chemistry and helix geometry. Remove the 2-hydroxyl and you get DNA. More stable, better for long-term storage, friendly, major groove. Keep it for RNA. Less stable, more versatile, right at the edge where strands can both pair and fold. The dogma's arrows are geometry's arrows disguised as information flow. Engineering rides the same rails. PCR, polymerase chain reaction, depends on predictable melting and annealing temperatures, which derive from base composition and helix thermodynamics. Sequencing technologies, Sanger, Illumina, Nanopore exploit how polymerases step along helices or how single strands thread through pores. Nanotechnology uses the helix as scaffold. DNA origami folds strands into shapes by programming where complementary segments are. None of this would be feasible if the helix were not programmable by sequence in a way that produces reliable shapes. Programmable matter is a fashionable phrase. DNA earned it by being a molecule whose macro behavior folds, stiffness, exposure, is a calculable function of micro choices, bases, and their order. Against all this order stands evolution's demand for change. The double helix accommodates change gracefully. Point mutations alter letters. If the change preserves helical shape at critical sites, the genome survives. If it improves a function, it is kept. If it harms, repair, or selection removes it. Repeats expand and contract through slippage. Transposons copy and paste. Recombination shuffles. Selection reads phenotype and writes back to genotype by culling or rewarding variants. Throughout, the helix keeps information copyable and readable while allowing diversity. The structure neither freezes nor flies apart. It is robust and permissive in just the right measure. What then does it mean to answer, why is DNA a double helix? At the end of this longer walk, it means acknowledging that reasons cascade. At the bottom are physical constraints. Water's hostility to exposed aromatic rings, the juice of pi stacking, charged backbones kept apart by ions, sugar stereochemistry steering bond angles, chirality, locking handedness. Those force a twist. Once twisted, new properties emerge. Grooves readable from outside, uniform width that equipment can fit, stiffness and flexibility in a Goldilocks zone, topological handles. With those properties in place, evolution invents a cast of proteins that exploit them. Polymerases for copying, helicases for opening, nucleases for cutting, ligases for sealing, topoisomerases for managing stress, histones for packaging, transcription factors for reading. Behaviors at the cellular level, replication timing, chromosome segregation, gene regulation flow from those inventions. Behaviors at the organismal level, development, memory, adaptation flow from cellular behaviors, and culture, our ability to diagnose, treat, edit, and design, flows from having learned the rules. From certain angles, the helix looks like inevitability. From others, like an accident. Both can be true. Swap solvents, swap base chemistries, swap temperatures, and perhaps another world invents a different scaffold for memory. But given our world, the double helix is not a coincidence. It is the favored solution among many, selected first by physics and then by the sieve of survival. Had life tried to store its blueprints in triplexes globally, replication fidelity might have choked. Had it persisted with single strands for large genomes, error would have overwhelmed. Two strands do what one or three would not. They let a code be self-complementary while staying easy to open, easy to proofread, easy to package. End not with awe alone, but with usefulness. When clinicians design a primer to detect a pathogen, they count on helix thermodynamics. When a scientist turns on a gene with a CRISPR activator, they count on major groove readability and helix rigidity at the target. When a cell survives sunlight by cutting out a dimer and patching the road, it counts on uniformity to spot the pothole. When a chromosome coils for mitosis and uncoils for interphase, it counts on twist as a knob. Each act is a small vote of confidence in a spiral staircase built billions of years ago. And so the question resolves. DNA is a double helix, not because someone preferred beauty, though the beauty is undeniable, and not because clever people declared it so, though clever people uncovered it. It is a double helix because a world of water and ions asked flat bases to hide and charged rails to show, because sugars held those rails at angles that favored a twist, because two strands together gave birth to complementarity and copying, because the twist created grooves that invited reading, because topology gave tools, because uniformity invited repair, because stiffness and flexibility balanced packing and access, because evolution took the gift and learned to use it. The ladder that twists is more than a picture in textbooks. It is a way that atoms hold hands to make meaning, a form that turns chemistry into memory, a geometry that lets living things write, read, and revise themselves. Out of all the shapes that flicker in the space of possibilities, this is the one that carried drops of chance toward the sea of continuity. We live because, long ago and then again every second, a pair of strands agrees to spiral.